So, when you picture genetics, you probably imagine those classic X-shaped chromosomes from biology class, right? Well, it turns out that one of the most common animals on the planet, the chicken, has a genetic blueprint that looks, well, almost alien compared to ours. Okay, let's get into this, because it's a really bizarre and fascinating story. And this is the big question, isn't it? Why would evolution go to the trouble of packaging a bird's genes in a way that's just so different from mammals? The answer, believe it or not, was hidden for decades, mostly because it was written off as totally unimportant. But as you're about to see, it holds a huge lesson about making assumptions in science. All right, so to really get your head around just how strange the chicken's genome is, we need something to compare it to. And the best comparison is us. So let's take a look at the basic genetic layout of a human versus a chicken. Okay, on the left, you've got the setup you probably recognize. The human genome has about 46 pretty large, easy-to-see chromosomes. Nice and simple. But then you look at the chicken on the right. Whoa. It has a few big ones, which scientists call macrochromosomes, but then it also has this swarm of over 60 tiny little dog-like chromosomes. They call them microchromosomes. And for the longest time, nobody really knew what to make of them. So, because these microchromosomes were so small and just plain difficult to study with the tools they had, this whole myth started to build up around them. The general consensus, the scientific wisdom of the day, was that they were basically genetic junk. And I'm not kidding, this was not some fringe idea. I mean, look at this quote from all the way back in 1955. For most of the 20th century, the official story was that these little specks of DNA were genetically inert, inactive, unimportant, just there. And they had their reasons for thinking that. It wasn't completely out of nowhere. They saw them as just evolutionary leftovers. Under a microscope, they all kind of looked the same. You couldn't tell them apart. They seemed to be made of this tightly packed, silent type of DNA called heterochromatin. And they couldn't find any of the usual genetic signposts on them. So all the evidence they had seemed to point in one direction. These things were totally insignificant. But then, 1998 happened. A team of researchers with some new techniques decided to take a much closer look. And what they found didn't just challenge the old myth, it completely blew it out of the water. And here's the best part. It wasn't just one single piece of evidence. No, they uncovered a trail of 3 separate, really powerful clues that all pointed to the same shocking conclusion. So their first clue was all about a chemical tag. You can think of it like a tiny molecular on-off switch that gets attached to DNA. Here's how it works. DNA that's turned off, that's silent, is hypoacetylated. It barely has any of these tags. But DNA that's buzzing with activity, packed with working genes, is hyperacetylated. It is just covered in these tags. So when the researchers took a look, they found that the supposedly inert microchromosomes were intensely hyperacetylated. They weren't silent at all. They were basically screaming with genetic activity. Okay, on to the second clue. This one is all about timing. See, when a cell divides, it doesn't just copy all its DNA at once. There's an order to it, and that order tells you a lot about what the cell thinks is important. It's pretty simple, really. The most important gene-heavy DNA gets copied first, right at the beginning of the replication cycle. All the less important stuff, the silent DNA, has to wait till the end. So the big finding? The microchromosomes were being copied right at the front of the line, along with all the other most critical genetic material. It was another massive sign that these things were anything but junk. And that brings us to the final clue, which was really the knockout punch. The researchers decided to stop looking at indirect signs of activity and just go in and try to count how many genes were actually there. So they looked for these things called CpG islands. They're really reliable markers that tell you a gene is nearby. And as you can see from this chart, the result was just stunning. The microchromosomes weren't just a little bit denser in genes, they were 6 times denser than the big macrochromosomes. These tiny dots were actually genetic hotspots. So with all this new evidence piling up, the activity markers, the early replication, the unbelievable gene density, it was pretty clear the old definition had to go. It was time to completely rewrite the book on microchromosomes. And this is the new reality. A microchromosome isn't junk at all. It's a tiny, super compact, hyper-efficient little genetic hard drive. It's designed to pack as much important information as possible into the smallest space imaginable. To put a number on it, the researchers figured out there's a gene roughly every 10,000 base pairs. Now, to put that into perspective for you, that level of density is right up there with the pufferfish genome, which is famous among biologists for being the gold standard of genetic compactness. And all of this builds up to the final, absolutely jaw-dropping conclusion. If you ask, so where are the chicken's genes? The answer is right here. A staggering 75%, 3/4 of every single gene in the chicken is packed onto these tiny little chromosomes that had been dismissed for decades. Now, this discovery wasn't just some fun fact for a biology textbook. It fundamentally changed how scientists looked at bird genetics, and it led directly to the development of some really powerful new research tools. You see, once scientists knew that all the real action was happening on these tiny chromosomes, they could finally build tools to analyze them properly. And one of the best examples of this is a modern online database called GEGA. GEGA, which stands for Gallus Enriched Gene Annotation, is a direct result of that 1998 discovery. It's this massive online resource that pulls together gene info from all the major international projects. It maps out which genes are active across 47 different chicken tissues, and it lets scientists easily dig in and explore the genetic reasons for specific traits. It's a total game changer. And this has huge real-world applications. We're talking about researchers using this knowledge to track down the exact genes responsible for things like epilepsy in chickens, or to identify the genes that control fat production, which is obviously vital for agriculture. It helps them understand the genetic basis of all kinds of diseases. All of this comes from finally appreciating those tiny, powerful chromosomes. For half a century, the vast majority of the chicken's genetic code was just hiding in what everybody had dismissed as junk. It really makes you wonder, right? What other profound biological secrets are sitting right there in front of us, just waiting for someone to challenge an old assumption and decide to take a closer look?